So I'm Katrina Whiteson. I'm here introducing Daria Van Tyne, who's joining us from the University of Pittsburgh. She has her education from Vassar College and also from Harvard University, where she did her PhD. I'm most familiar with her work when she was a postdoc with Michael Gilmore and got working with Enterococcus. One of the figures she generated is one of my favorite figures of all time, showing the history of Enterococcus being associated with hosts back to the split between nematodes and sponges. So it's a very ancient association. And I'm just really looking forward to getting to hear about her work with Enterococcus and phages. And so I don't want to take more of your time. I'm really looking forward to hearing about it. And thanks so much for coming all the way to share your research with us. Thank you.
Thank you. Thank you so much for that kind introduction. Thanks for letting us know about the audio. I would hate to have me talking to all of the people joining remotely. And you just see me talking and not hear me. So it's really a delight to be here with you all. Thanks to iPath for the invitation. Thanks to Lizzie for her help coordinating the visit. I gotta say it's been seamless. And I feel really lucky to be here sort of in your presence and to have the opportunity to tell you about some of the work that is ongoing in my lab and how it relates to phage therapy. So I thought by way of introduction, I would just tell you quickly about the various research interests in my lab. We're not just a phage lab. I didn't train in phage, and I feel like kind of a poser in the presence of phage biologists. But the research in my lab is broadly divided into three categories. We do a lot of work using functional genomics approaches to try and understand how drug-resistant bacteria are evolving in people during infection. So we isolate bacteria from patients with bad infections. We sequence the genomes of the bacteria. We compare the genomes to one another to try and generate hypotheses that we can then go study in the lab. And a lot of the work revolves around evolution of antibiotic resistance or bacterial evasion of host immune defenses. A second research interest in the lab is how mobile genetic elements move between bacteria in the hospital. So we work with epidemiologists who sequence the genomes of bacteria from hospital-acquired infections, and we study how things like plasmids and other mobile elements move between bacteria in the hospital. And then finally, we're interested in phage therapy, and that's what brings me here to you all today. So we're exploring how phages might be able to be harnessed to treat antibiotic-resistant infections and trying to add new tools to the antibiotic armamentarium toolbox, if you will. So I'm not going to spend too much time on this slide because I think we're all pretty like-minded in thinking about phage and phage therapy. But my real interest in phage therapy is driven by the sort of clinical need for new tools to treat resistant bacterial infection. So we know that antibiotic resistance and multi-drug resistant bacterial infections are a looming threat to public health. And obviously bacteriophages are viruses of bacteria. I put up the phage life cycle here just to remind us that, you know, bacteriophages can adopt two different lifestyles, and I'm actually going to talk about both in the talk today. From a clinical standpoint, we're most interested in the lytic ability of phages. They can target bacterial cells for killing, but we also want to remember that prophages can integrate into the bacterial genome, and this lysogenic cycle is also sort of going on in the background for some phages. So obviously we put the two things together, increasing rates of antibiotic resistance, also decreasing interest in the development of new antibiotics have prompted this renewed interest in phage therapy. And of course, I put this slide in. I don't have to tell you all that there's a phage therapy center here at UCSD, but when I talk to other people, I like to remind them that phage therapy centers are starting to form and that the field is relatively nascent, but clinical trials are underway. And it's really exciting to be in this, this really developing field. So the outline of the talk today, the overall theme of the talk is I'd like to tell you about how we in my lab use cases of clinical phage therapy as a starting point to do what we do, which is functional genomics research. And I'm going to tell you about two different patient cases. The first one was where we used phage therapy to try and treat a patient with refractory Burkholderia multivarians infection. And then the second part was a separate patient that we treated with phage therapy for recurrent Enterococcus faecium infection. And in both cases, I want to tell you a little bit about the patient case and how we used our sort of functional genomics expertise to try and learn about what happened during the case. And then in both parts, I'm also going to tell you a little bit of the follow-on work. that we did that was largely inspired by the experience with the patients. So how does phage therapy work in our setting? So I'm at the University of Pittsburgh, and we're affiliated with the University of Pittsburgh Medical Center, UPMC. So the way phage therapy works in our setting is that my lab over the last couple of years has isolated phages against various organisms from various environmental sources. In all of our screening, though, we have used clinical isolates that represent the types of bugs that we might like to treat. And so my colleagues in the Division of Infectious Diseases have collected all sorts of drug-resistant bugs from all sorts of bad infections, and we use those isolates to screen for phages. And then we collect wastewater either from Pittsburgh area hospitals or the municipal water system in our area, and those have been our primary source for phages. So we do all this screening, and we assemble libraries of phages. And then as sort of word travels and physician colleagues know that we're doing this work, they start referring patients to us. So we receive clinical isolates from patients at UPMC, where the physician identifies the patient as a potential candidate for phage therapy. We screen the isolates against our libraries of phages. If we find a match, then the active phages can be purified and formulated. We've done this in my lab. We've also worked with other labs to do this. We go through the necessary regulatory steps, both with the FDA and with our local IRB. And when they all approve and give the green light, then we can give either the single phages or phage cocktails to patients. And in all cases, the patients have also received antibiotic. So phage has been an add-on therapy for all of these patients. And of course, after we treat them, we monitor the patients for things like safety, for the clinical response, for the development of phage resistance, for antibody responses. And so far we've been involved in treating 7 different patients at UPMC. I'm going to show you a table on the next slide that sort of goes through the details for each case. But I just want to stop for a second and acknowledge the many people that it takes to do this work. So my clinical collaborator, I'm a PhD, I'm not a physician. And so to do this work, I work with a bunch of physicians, but Gadi Haidar is my sort of partner in this work. Madison Stelfox is a postdoc in my lab. Ryan Shields is a pharmacist who helps us get isolates and also think about dosing, pharmacokinetics, pharmacodynamics of phage. Kaylee Hughes-Kramer single-handedly does all of the INDs for us and is just fabulous. And then these guys, Ben Chan, Brecht Wehrkopf, and Austin Terwilliger have helped us either find phage or prep phage for patients at various times. And the first case I'm going to talk about, the phage is actually, the phage came from Ben Chan. The second case, the phages came from Brecht Wehrkopf. So this is the table that summarizes all the patients that my lab has been involved with treating with phage therapy. This is sort of independent from my colleague Graham Hatfull, who's also at University of Pittsburgh and basically has the market cornered for mycobacterial phages. And basically mycobacterial cases go to Graham and we kind of take everything else. And so these are the cases that we've been involved in. And I'm going to tell you about this patient number 2 up here that received IV and inhaled a phage called a BCH7. And then also the third patient here that was a patient with recurrent EPC and bacteremia that we treated with phage therapy. And as you can see, lots of different organisms, lots of different phages. They came from lots of different places. My lab has either provided the phage or facilitated the phage matching and the administration in all these cases. And as many of you probably already know, we have not seen any adverse effects of phage therapy. We determined that it was safe in all the cases that we've treated so far. So to get right to the first patient, this was a 32-year-old female with cystic fibrosis. She had known both respiratory and sinus colonization with Burkholderia multiforians going back since at least 2001. She received a double lung transplant in 2017, and then in the fall of 2020, she was in the hospital with pneumonia and bacteremia with multi-drug resistant B multiforians. So this is a summary, a very detailed, busy summary of her sort of clinical course that we were involved with. And so at the very top on this timeline, we're showing the respiratory cultures and blood cultures. All of the red boxes are B multivarians that were growing in these cultures. And then the timeline is divided into the time before she got phage therapy and then after she started therapy. She received about a week of inhaled phage therapy before she ultimately passed away. And she was on basically anything that she could be on in terms of antibiotics. And despite all of this antibiotic therapy, she was persistently bacteremic with this Burkholderia multiforians. So the phage search was initiated here in the timeline, and Ben Chan is a Superman, and he worked like through the night to try and identify a phage that was active against her isolate. And so in just 10 days, he received her isolate, he screened it, he found a phage called BCH7, and he formulated it for therapy. So she started her inhaled phage therapy here, and like I said, received inhaled phage therapy and then actually a single dose of IV phage therapy before she ultimately, unfortunately, succumbed. So we were involved in the patient starting here, and of course, they were admitted to the hospital, and so we collected what we were able to collect in terms of like remnant and clinical specimens from the patient. And so we were able to collect respiratory specimens, which were either tracheal aspirate or bronchial alveolar lavage fluid, so samples from the patient's airway during the course of her therapy. And so we used quantitative PCR to look for changes in bacterial abundance as well as phage abundance in these samples over time. And so what these graphs are showing are qPCR quantification of either Burkholderia multivarians DNA or phage DNA in the same samples over time, and they're ordered, throughout her course of therapy. And what we saw was that looking at the bacterial abundance, there was a small decrease. It was significant, but it wasn't a huge drop in the bacterial abundance in these samples over time. What was very interesting to us was that when we looked for phage DNA, we actually didn't see any phage DNA until the very final time point. So on the day 6 time point, we suddenly observed a decent amount of phage DNA. And we wonder about this. Is it because it took this long for the phage to find its target and start amplifying? Or was there something else that prevented us from really seeing or detecting phage DNA beforehand? But we know nothing about the dynamics of phage replication in vivo in a critically ill patient. And so I think the fact that we saw phage even at one time point was striking to me. And then finally, we had serum. We got blood samples from the patient so we could look for evidence of a serum neutralizing response. And we found no evidence of serum neutralization. So the patient's serum was unable to neutralize phage, before her therapy, and as well as after 7 days of therapy. This is probably too early in the course to even see a neutralizing antibody response, but we nonetheless tested and found that there was no evidence of phage neutralization from her serum. And so at the same time, we were collecting bacterial isolates from the patient. We also contacted John LaPuma at the University of Michigan, and he had historic isolates from the patient. So he provided us with a couple of historic isolates, and we had these contemporary isolates as well. So we sequenced the genomes of 15 different isolates that were collected beginning in 2001 through the 2020 hospitalization and phage therapy course. So analysis of the bacterial genome sequence data showed that the isolates collected in 2001, the historic ones, were closely related with the contemporary ones. And this is consistent with other studies of pathogen evolution in CF patients. Most CF patients become colonized and infected with a single strain that evolves with that patient over years. And so that was we sort of confirmed that she had this same organism since 2001. And this phylogenetic tree shows the 15 different isolates that we sequenced. And next to the phylogeny is the susceptibility of each isolate to a variety of different antibiotics, as well as to the phage, BCH7, that we used for therapy. Now, the striking thing here, which was unexpected to us, was that we found that the two oldest isolates, isolated in 2001 and 2003, were actually resistant to the phage that was used for therapy. So these historic isolates were not susceptible to the phage. And we think that something happened over time of pathoadaptation of this bacteria to this host that rendered the bacteria susceptible to the phage. And this is just showing with a plaque assay that this earliest isolate from 2001 was resistant to phage. So here we've spotted serial dilutions of phage onto the bacterial lawn, and we don't see any evidence of growth inhibition. And that's in contrast to one of the contemporary isolates where we do see evidence of phage activity. We looked in the genomes and we found some mutations in genes predicted to impact LPS biosynthesis. And in fact, we extracted LPS from the historic isolate and the contemporary isolate, and we saw these high molecular weight bands correspond to the O antigen repeats in the LPS. And so it seemed like the contemporary isolate mutated its LPS to remove O antigen, and in doing so, it became susceptible to this phage. And this raises issues about, for patients like this, when is the right time to look for phage? If we had started a phage hunt for this patient back in 2001, we may not have found phages, or we may have found different phages. from the one that ultimately was active against her isolate at the time that she was ill. So this is where this kind of study ended, and this is a case report that's now under review for publication. But to take this work kind of forward and think about what the next steps might be, we've been thinking about, how could we find more phages for an organism like Burkholderia? And so this kind of seeded a larger project that was undertaken by a technician in my lab who was also a master's student. And so I'm going to tell you some of the highlights from Haley Nordstrom, who's down here on the right. This is her master's thesis work that she completed in my lab last spring. So, patients with Burkholderia infections, sometimes these infections are very difficult to treat. These Burkholderia bacteria are environmental organisms. They're intrinsically quite resistant to antibiotic. And so here's a case where if you could find phages for these organisms, you have the potential to make a difference for some of these patients. But it's very difficult to find phages for Burkholderia. This is a genus of many species that are super diverse. They don't grow well in the lab. They're super slow growing, and they're understudied. We don't know a lot about their biology, much less their phage biology. So we've collected and sequenced about a little over 30 Burk clinical isolates from UPMC patients. And when we look in their genomes, we find that each Burk isolate genome has between 1 and 7 prophages that are predicted in the genome. And so we wondered, could this be a source of phages? The genomes of these clinical isolates that have all these prophages in them, could this be a source of phage for targeting Burk infections in patients? So our collaborators in this work are Lee Harrison, who's an epidemiologist and does routine whole genome sequencing of possible acquired infections. And it was from Lee's lab that we got many of these clinical isolates and their genomes. And then Katie, Marissa, and Batsala are members of the Harrison lab that assisted in this effort, both isolate collection, genome sequencing, and then genome analysis. So to just take a minute and step back and remind ourselves about the difference between lytic and lysogenic phages. So whether a phage is lytic or lysogenic largely depends on the phage as well as on the bacterial host. So phage, the same phage could adopt both lifestyles. But if we think about prophages, these phages that are integrated into the genomes of bacteria, these are the result of successful phage infection. So a prophage, that is a phage that was successful at infecting that particular host at some point in its evolutionary history. So we think that prophages might be a useful starting point for getting phages that we could then modify and engineer further. And of course, you probably are all familiar with this approach from Graham Hatfield's work, where they have in Mycobacterium taken prophages and engineered them to make them obligately lytic. So we're trying to explore, whether the same approach could work in the case of Burkholderia. And so this is basically what Haley did for her thesis project. So she took 32 clinical isolates of Burkholderia, and she induced prophage release by simply treating the bacterial culture with mitomycin C, which we were hoping would induce some of these prophages to excise and amplify themselves. And then she took the prophage-containing lysates, filtered them so that we have bacteria-free but phage-containing lysates. And then she spotted each lysate onto a lawn of Burkholderia bacteria. So she did a screen of 32 different phage-containing lysates against the same 32 Burk bacterial isolates. And she was looking for pairings where the lysate would inhibit the growth of the indicator strain, the target strain. And so she started in this large screen. There were 1,024 pairwise combinations of a source lysate with prophage in it versus a target clinical isolate. And from those 1,000 or so combinations, it turned out that there were 11, small number, but there were still some. There were 11 pairings where the lysate would inhibit the growth of the target isolate lawn, so about a 1% hit rate. And every time she saw a hit, she would follow up to confirm that the inhibitory activity was actually due to phage. So she would take the lysate that inhibited growth, she would serially dilute it, and then she'd look for plaques. And if she saw plaques, she would pick and passage those plaques on the same strain. So in doing this with these 11 pairings, she ended up with 7 that sort of made it through this follow-up period. And so she ended up with 7 isolated phages from this work. And through genome sequencing and additional testing, we found that 4 of these 7 phages represented 4 non-redundant, 4 new phages. So Haley did all this rigorous screening, and I'm really thankful to her. She was still smiling at the end of it. And yeah, it was a big effort, but she took it on with a smile. So now that we had these 4 prophage-derived phages, we wanted to look at their spectrum of activity. And so we did a host screen of the 4 isolated prophage-derived phages plus 2 additional Burke phages. And we tested the panel against 35 different Burkholderia clinical isolates. So the result of that screen is shown here. And so to just orient you, here are all the different bacterial species we tested. So these are all Burke clinical isolates. Most of them are in the Burkholderia cepacia complex, which is basically this branch of the phylogeny, everything except for this B gladioli up here. And most of the isolates that we collected were either Burkholderia multivarians or Burkholderia senocepacia. These were the 2 primary species we sampled. And then the phage susceptibility results are shown here. These 4 BCC phages are our prophage-derived phages, and then we had 2 additional phages. And this BCH7 was the phage used for therapy in the patient that I talked about. So we see that, each phage has variable activity. It could target STU as 3 isolates, as many as 12 different isolates. Most phages were active against isolates belonging to different species. So the phages are kind of picky in their host range, but they many of them were able to infect species belonging to different, isolates belonging to different species. This BCC02 prophage-derived phage was the most active phage that we tested. So you can see that it could target a large number of isolates here. And we did some electron microscopy and confirmed that it was a legit phage, right? It was making intact virions and had the predicted morphology. And one other thing that we noticed from this sort of host range screen that we did was that most of the senocepacia isolates were resistant to, every senocepacia isolate was resistant to 1 phage in the panel, but there were 8 B multivarians isolates that were resistant to all the phages that we tested. And so this effect of we compared the multivarians versus the senocepacia, and we found that the multivarians isolates are more phage resistant compared to senocepacia. And then when we looked in their genomes, we also noticed that the multivarians isolates had more prophages in their genomes compared to senocepacia. And we wondered if this might explain, in part, their reduced phage susceptibility. So we looked a little bit more closely at the actual prophages that these isolates were all encoding, and we compared prophage genomes with one another. And we identified one particular prophage that we called B multi PP1 that was found in the genomes of 10 different B multivarians isolates, and that group of 10 included all 8 of the phage-resistant isolates. And so to just zoom into the B multivarians part of the host range screen here, we see in general the species itself is pretty resistant to phage, but then the isolates themselves that had this particular prophage PP1, 8 of the 10 with the prophage are totally phage resistant. And so this is an interesting correlation. We obviously don't know if it's causative, but it's at least a place where we could explore more around the genetic determinants of phage susceptibility in B multivarians in particular. So to wrap up this first part of the talk, we started with this question: could prophages from one Burkholderia clinical isolate lyse other clinical isolates? And the answer is yes. Our screening hit rate was pretty low, but it still yielded 4 active phages that were new phages to us. This prophage-derived phage, BCC02, seems like the best hit from this screen. It had a pretty wide host range, and we think it's the best candidate for genetic engineering to follow on and try and make it obligately lytic. And this prophage, B multi PP1, was strongly associated with phage resistance, and we wonder if this is correlative or causative. And I wanted to stop here before I switch gears and talk about Enterococcus. And I'm going to drink some water, and if there are questions, I'm happy to take a couple questions before I do the other part of the talk. Yeah. Oh, wait, sorry. We're going to get a mic to use just so that the people in the Zoom land can hear your question.
So I think it's really neat that you can get prophages out, but don't they continue to lysogenize the strains that it looks as if they infect? And then isn't that risky because many temperate phages will carry genes that enhance the virulence, or at least the vigor of the bacterial strain that harbors them?
Certainly. Yeah, it's a great question.
Even if you select clear mutants.
Yes, certainly. So, yeah, that from a therapeutic standpoint, prophages are certainly not the preferred option for giving a patient phage therapy because of the reasons that you just articulated. We view it as a potential source of raw material of phage that we could then engineer to remove their prophage potential. Whether that could happen or not, and the appropriate methods for doing that is still up for debate.
You can get multiple lysogenization.
Mm-hmm.
My understanding is that many Bacteria species or strains are soil organisms.
Correct, yeah.
What if you looked, as in Graham Hatfield's study of mycobacteriophages, what if you took soil samples and just selected?
Certainly, yeah. So we have looked a little bit. I have a farm share, and I sent some of my techs to the farm they grow my vegetables, and we took some soil samples, and we maybe didn't sample at the right time or didn't pick the right environments, but that effort was largely unsuccessful. I will say, because these bugs are soil dwellers, they really do not like growing under standard laboratory conditions. They do not like growing at 37 degrees. They do not like growing in relatively rich media, which is kind of funny because they cause infections in people. But there is certainly a mismatch between, our kind of standard growth conditions aren't well suited to the biology of the organism, and how that also impacts the phage bacteria relationship. Perhaps there were phage in those samples and we didn't get them because our growth conditions were not favorable to the phage.
Thank you.
Okay.
So on the same front as the conditions with the prophages, I'm just really curious, in our hands, prophages tend to be kind of delicate. Like they're so used to being in a more protected intracellular situation that they're like, you can't even freeze them. You can't wait a day. So how is that going for you guys working with them?
Yeah, so Haley actually left my lab over the summer to move to Maryland, and a new technician in the lab has picked up some of the work. And yes, they are kind of like delicate flowers. They need some careful handling. And we haven't tested like sort of long term their stability. I think that if nothing else, this work again kind of gives us a starting point to think a little bit more broadly about like what would make a good Burke phage. And the fact that we saw a broad range of lytic activity for this one particular phage suggests to me, at least, that might be a good starting point to follow on from, in terms of just thinking about like what are the genetic characteristics or the building blocks for a good Burke phage. I'm certainly not suggesting that we want to use any of these isolated prophages for therapy because of all the things that we're talking about. But in the absence of any phage, any other phages, I think it's a useful place to start. Yeah, maybe one more question.
Just sort of following up on the prophage trail as well, and it's kind of nice that you get to see this correlation that there's a lot of phage-insensitive strains that don't respond to your Bacteriophages just because there are prophages in them. I don't know if you've mentioned this, but have you sequenced them to see how similarly related they may be, just because there could be some sort of superinfection exclusion that might be playing into role, like why they're insensitive?
Certainly, yeah. So the multivorans isolates that have this prophage are not, they sort of spans the phylogeny. It spans the phylogenetic tree of Burkholderia multivorans. So it's not one specific clade. This prophage itself, it's not the identical same prophage in all of them, but it's the prophage sequences are closely related enough that we could call them the same. And we used 90% coverage and 90% nucleotide identity to say it's the same in our analysis. There are certainly differences between their sequences. And the hypothesis to test would be exactly what you said, that perhaps this particular prophage provides superinfection immunity to the strains that have it. And I think that's a testable hypothesis. Okay, I'm gonna move on to tell you now about Enterococcus. So the second patient I wanted to tell you about was a patient with recurrent EFCM bacteremia. So this was a female patient in her late 50s, and then she had recurrent episodes of EFCM bacteremia starting way back in May of 2013. Every year, every couple of years, she would have an EFCM bacteremia that was managed okay with antibiotics. She would clear the bacteremia and go home, but then, at some time later, she would recur, and it was always EFCM. And then she came to our attention in the middle part of 2020 because these infectious episodes, the recurrent infections kind of accelerated, and she ended up having 7 different hospitalizations in 2020 for EFCM bacteremia. And so we started following her in June of 2020, and that's observation day 1 on this timeline. So it's a very long timeline, over a year that we followed this patient. But her in the setting of increasing frequency of recurrent infections, she was sort of referred to our lab, and we started looking for phages for her. And we found one particular phage, which was called 9184 and was provided by the Dwerkoff lab at University of Colorado. In December of 2020, this patient was in the hospital and was bacteremic for 4 weeks straight, despite receiving IV daptomycin and vancomycin. And so we started her on phage therapy in December of 2020. And surprisingly to all of us, her bacteremia cleared immediately. So the only thing we changed in her therapy was to add this single phage. And after a couple of doses of IV phage, her blood cultures turned negative, and she was able to go home from the hospital about a week later. And so she continued to receive phage in the outpatient setting. This patient got both IV phage as well as oral phage. And she experienced a couple of recurrences of bacteremia. So bacteremia episodes E, F, and G happened during the time she was on phage therapy. She had negative blood cultures in between those. So they were real sort of recurrences, independent episodes. But she had a couple of recurrences of bacteremia in early 2021, and that actually prompted us to look for another phage, thinking that maybe if we gave her a cocktail, we would do a better job at suppressing the bacteremias. So we added a second phage to her therapy, and the second phage was called High 3. And so starting in April of 2021, she switched from a single phage to a 2-phage cocktail, and this was maintained for about 100 days more until the summer of 2021. And this final recurrence, I'll tell you why we decided to withdraw therapy after this final recurrence. So this clinical course is a typical phage therapy clinical course where there's a ton of antibiotics, there's a lot of stuff going on, but here we're summarizing the course of phage therapy. She was receiving IV daptomycin basically throughout, and then a smattering of other antibiotics, basically trying to suppress these bacteremia episodes. So we again collected isolates of EFCM from both the patient's blood, from all these bacteremia events, as well as from her stool. We sequenced a bunch of isolates, and we confirmed that the likely source of bacteremia for this patient was the GI tract. So she was colonized in her GI tract with the EFCM at pretty high abundance that was causing her bloodstream infection. We also were able to find 2 prior isolates that had been collected from her a year prior to the sort of in the phage therapy time course from VRE rectal swab surveillance. So we compared the genomes of all of these isolates from the same patient, and we confirmed that they are closely related to one another. This SNP accumulation plot is just showing a steady increase in SNPs relative to the first isolate. And it tells us that all of the isolates causing her bacteremia, as well as the isolates in her stool, were all part of the same population that she had been chronically sort of colonized with this whole time. And I will say at the time, we didn't have this genome sequence information, but we knew that she had both vancomycin resistant as well as vancomycin susceptible EFCM. And a big question from the clinicians was whether these were 2 different strains or just 2 different versions of the same strain. And it turns out that it was just 2 different versions of the same strain. So there was this EFCM population of closely related organisms, and some of them had the van resistance operon, and some of them didn't. So the patient actually also, I should say, also we knew that she had E. faecalis colonization. So we recovered E. faecalis from her stool as well, but she never had E. faecalis bacteremia. So there was something special about the EFCM that I still don't know what it is. But in this patient, it was either the bug or the patient. She was unable to clear EFCM from her bloodstream. So this patient got phage therapy, she got IV therapy, she also got oral therapy. And so we were really interested in investigating what was going on in her GI tract. during the course of her therapy. So this patient was really great. She was in the outpatient setting, but she provided weekly stool and also weekly serum samples that we took into the research lab to study further. So we asked this question of what's happening in her gut. And rather than doing shotgun metagenomics and looking at everything, we decided to target, like sort of train our shotgun on just the population of Enterococcus in her gut. So we took her stool samples, we plated them on a selective media called bile esculin azide agar, and this only allows growth of Enterococci. So we plate the stool on Enterococci, or on BA agar, and then we pooled the colonies that grew on this agar. So we made a pool of between 100 and 1,000 colonies that we thought were all Enterococcus colonies. We extracted genomic DNA from the pool, and then we did shotgun metagenomics, but again, only on the enterococcal population in her gut. And we did this work in collaboration with Brecht Dwerkhoff and a postdoc at the time in his lab, Mike Mangalea. And so the results of that effort are shown here, and I apologize, it's another busy slide, but I'll leave it up for a while and talk you through it so you can take it all in. So this is another time course. The bars on this graph are showing the time points of stool samples that we collected from the patient, and the colors, hopefully you can see the green or the gray versus the pink. So the gray is the relative abundance of Ephicalis in that stool sample, and the pink is the relative abundance of Ephesium in that stool sample. So we see some differences over time. There are periods where Ephesium was the dominant Enterococcus in her stool, and there were periods where Ephicalis was the dominant Enterococcus in her stool. And then we're also looked at the percentage of reads that mapped to the Van operon as a surrogate marker for the VRE abundance. And it was only Ephesium that we ever saw the Van operon in. So the VRE, the Van A operon read abundance largely tracks with the Ephesium abundance over time. Now at the top here, overlaid on top of each one of these time points is what antimicrobial therapy the patient was on at the time. So, as we showed before, she was getting daptomycin IV throughout this time frame. The phage therapy, when she got the single phage, high 3, is here shown in blue. And then when she switched to the 2-phage cocktail, is to the right here. And then she also received a short course of oritavancin, and as well as a single dose of tigecycline. And what I want to draw your attention to here is when she started phage therapy, she started IV and oral therapy, but it seemed like on the single phage, nothing really happened in her gut. So her gut still remained sort of monoculture of monoephesium. In early 2021, she received oritavancin, and it seemed like this did have a little bit of an impact on her GI enterococcal population. So the oritavancin seemed to drive down the abundance of Ephesium and allow the Ephicalis to sort of come up in number, or at least, relatively we started to see Ephicalis again. But that was short-lived, and Ephesium kind of came back again. And then when she received this tigecycline, it again seemed to precipitate a shift away from Ephesium towards Ephicalis. And we wonder if the sort of addition of this high 3 phage to her therapy may have helped prolong this period of Ephicalis domination in her gut and helped suppress her Ephesium GI population. And we see this is also the time in her course when she was bacteremia free. So during this time, the patient stayed out of the hospital. She was able to go on vacation with her family, which she had not been able to do in the preceding years. And so during this time in her therapy, whether it was due to phage or not, she definitely experienced an increased quality of life. And then to just draw your attention to this final recurrence, bacteremia episode G, we see actually that prior to this recurrence, her preceding stool sample had an increased abundance in Ephesium. And this actually corresponded to her going off daptomycin. So she stopped taking daptomycin, tried suppression with tadazolid and doxycycline. And it seemed like that removing the daptomycin may have, driven up the Ephesium again. So this is basically what we had in terms of metagenomic data from her stool, but we again had isolates throughout the course of infection. And so one thing we asked, is when she had bacteremia after getting phage therapy, did the bug evolve resistance? And so we tested all the isolates that we could during that came from her blood during phage therapy. And we saw that there was really no evolution of phage resistance over time. So even isolates from this final bacteremia event, episode G, these 2 isolates were from that time, and they remained just as susceptible to both phages as all the prior isolates, even before she received phage therapy. So we saw no frank evolution of phage resistance in the bugs from her blood. And this is despite a very long treatment duration, and was notable to us. The other thing we did was look for evidence of serum neutralization of phage activity. And so to do this test, we simply take serum from the patient and we incubate it with phage overnight. And then we look for evidence of phage activity. And we compare activity of the phage plus serum condition to the phage plus buffer condition as a control. And if there is serum neutralization, we would see that phage plus serum has less phage activity or no phage activity compared to phage in the presence of buffer. So we can use these 2 values to calculate an efficiency of plating. And when we do that for serum samples taken throughout the course of this patient's treatment, we found that surprisingly, but maybe not surprisingly, when we switched the patient to a 2-phage cocktail, about 6 weeks after starting that 2-phage cocktail, we detected neutralizing activity in her serum. So the whole time she was receiving the single phage at 1 × 10⁹ PFU per dose, there was no big change in phage activity in the presence of her serum. But after we switched her to a 2-phage cocktail, we see a pretty precipitous decline in phage activity in the presence of her serum, and what looks like the development of a phage neutralizing response. And Maddie Stelfox took this further to look for an IgG specific response using a custom ELISA assay, and was able to find that, that there was indeed evidence of increased phage specific IgG during this time period when we saw phage neutralization. So it seems like in this patient, it was either adding the second phage or doubling the phage dose triggered this neutralizing antibody response. And so that's all I wanted to tell you about the case. And when we saw the serum neutralization, and we and the clinical team and the patient decided to withdraw therapy and to just close the clinical case, the patient remained sort of at home, but was chronically ill and ended up passing away about a year after finishing phage therapy. And so to just finish up with the last part of the talk about sort of where this has led us on the research side. So these 2 phages that were used in this patient are both phages, and the Dwerkoff lab has characterized them a little bit with genome sequencing. They are distinct phages. They're totally different from one another. It appears that they use slightly different receptors on the bacterial cell surface, both polysaccharides, but the 9184 phage appears to use the capsular polysaccharide, whereas the high 3 phage appears to utilize a different polysaccharide on the surface for infection. And so we at our hospital have a lot of patients that have VRE, both colonization as well as bloodstream infection. And we wondered about the clinical utility of these 2 phages. So we found them as being active against this patient's isolate, but we wondered about all the other patients with VRE infections in our hospital. So we simply asked, how active are these 2 phages against the VRE that cause infection in other patients? And we also wondered, because these 2 phages are different and they seem to use different receptors, is their activity correlated with one another? And could they work synergistically? Do they work antagonistically? We knew nothing about this when we treated the patient, but we're curious about the utility of these phages more broadly. And so Maddie most recently has screened these 2 phages against 89 different VRE isolates collected from 75 patients that had VRE bacteremia bloodstream infection between June 2020 and February 2022. So this is like a contemporary set of isolates from our setting. So she tested both phages alone, so just by themselves, as well as in a 50-50 cocktail to ask, are they active? Is one phage active? Is the other phage active? And are they active together? And then she looked for trends in activity by sequence type. And I'll just say ST sequence type is a way that we kind of keep track of the genetic lineages of the epithelium in our setting. So this pie chart is showing the distribution of different sequence types of different types of VRE in our setting. And there are a couple of groups, a couple of lineages that are more dominant, but it's pretty heterogeneous in terms of the genetic makeup of the epithelium population in our setting. And these isolates were not cherry-picked in any way. It was just sequential VRE blood cultures. So we think this is pretty representative of at least our local epidemiology for this bug. And here's the result. I apologize that it looks a little blurry, but this is a phylogenetic tree of all 89 Ephesium isolates. And the colored rings on the outside show the susceptibility to either the 9184 phage, which is in red, the high 3 phage, which is in blue, or the combo of the two phages together in purple. And so the things that stand out are just at the broadest sense. 71% of these isolates were susceptible to either phage alone or in combination. So about 2/3 of the time, the phage was active against the isolate that we tested. There were a couple of sequence types, a couple of lineages that were largely phage resistant, including ST18, who are all over here, ST80, who are down here, and ST412, who are over here. And we wonder what it is about these lineages that makes them resistant to these phage. Perhaps they have different versions of the capsular polysaccharide or different EPA antigens that are not recognized by the phage as receptors. We saw that the activity of the two phages was very strongly correlated. So most of the time, if an isolate was susceptible to one phage, it was susceptible to the other. There were a couple of cases, 7 cases, where there was differential susceptibility. And in all those cases, the bacteria were susceptible to 9184, but not to high 3, which I think is kind of interesting. And then looking for synergy and antagonism, there were 3 cases where we didn't see any activity of either phage alone, but the combination, we saw activity. And then there were 6 cases of antagonism where either phage alone, we did see activity, but the combo together, we didn't. But again, most of the time, if the isolate was susceptible to one phage, it was susceptible to the other phage, and then it was also susceptible to the combination. But with this information, the next steps, and I'm not going to tell you about that today because the work is ongoing, but with this data, right, a large collection of sequenced isolates and this phenotype data, we can now go into the genomes and try and identify and study potential markers of phage susceptibility and resistance. And hopefully this is going to teach us more about the biology of these phages, but also hopefully inform our phage therapy efforts going forward. So that's all I wanted to tell you about today. In conclusion, we think that phage therapy is useful for treating drug-resistant infections. It's desirable because it's very highly personalized and as a targeted therapy. We think that phages in many cases, but probably not all cases, could synergize with antibiotics. We're working to build a phage therapy program at Pitt and UPMC. We don't have a name for our phage therapy program, but we did organize recently under this Pittsburgh Phage Project, which was just a small grant that we got to sort of get together all the phage researchers in the Pittsburgh area. And if you're interested, you can visit the website for the Pittsburgh Phage Project and learn more. But in my lab, we use these experiences with the phage therapy cases to inform our research going forward. As a PhD and a researcher, we service the clinical phage therapy efforts, but we work in the research lab. And so we want to try and treat each case as an opportunity to learn more about bacterial evolution, about phage biology, and about how to do the next case better. And of course, clinical trials for various cocktails targeting things like Pseudomonas and staph are underway. But at least in our setting, there's definitely going to be a need for phages for other organisms for a long time, including the bugs I told you about today. And just quickly to put a plug in for Achromobacter, we're actively working on a pilot Achromobacter phage therapy trial. And I want to thank all the members of my lab who actually did all the work that I told you about today. I thank you all for your attention, and I'm happy to take more questions.
Hello, I just had a question as to whether you guys sampled for any active infectious particles that was coming out of the stool to verify that it made it to its target.
Yeah, we haven't yet, but we saved stool samples from the patient. And so that's one thing that we could do either by looking for active phage or using qPCR to look at phage abundance. I'm inclined, I was talking to someone this morning about just filtering, taking the stool, filtering it for the, viral fraction, and then doing shotgun sequencing. And also we have the potential to look for any evidence of adaptation of the phage. The important caveat there, though, is that patients that get phage therapy, at least in our setting, they get 2 doses a day, 3 doses a day of the parent phage. And so our ability to observe evolution might be hindered by the fact that there's a constant reseeding of the system with the ancestor. But we have the samples and we can at least look and see what we can see.
Hello. Out of curiosity, do you happen to know how the phages were orally administered and whether they can like stand up to the environmental conditions?
Yeah, it's a great question. So this patient was on a proton pump inhibitor, and so we had her drink an oral suspension of the phage, basically right after she took her proton pump inhibitor. Yeah, whether any active phage actually made it into her gut or not, we have the samples though, so we can look and test, but it is definitely an open question. I will say Intralytics, this company, is doing like a VRE phage therapy trial with oral phage. And I talked to the CEO there, and he's like, we're just doing it the Georgian way of, some sodium bicarbonate, drink some sodium bicarbonate, and then drink the phage, and just like cross our fingers and hope. It's an open question whether you should encapsulate phage in nanoparticles or capsules. I think those are all open questions and in need of exploration, and probably that needs to happen in the research lab.
I was wondering whether it was like in some kind of like starch capsule or in a liquid nanoparticle or.
So people talk about this all the time, but nobody's actually done the tests to see if that would help, with stability and help with getting the phage where we would like it to go. Yeah.
Thank you.
Hello, everyone. So nice talk. I was wondering if like fluctuation of like efficient versus efficacy. You said that whenever there was like decrease in efficient, the patient was feeling better.
Mm-hmm.
So I was wondering if like efficacy doesn't have any like pathogenic effect on the patient.
Can you say the last part again? Sorry.
Yeah, like when the efficient was kind of lower,
yes,
there was efficacy was increased, right?
Yeah.
But you said the patient was feeling better during that time.
Certainly, yeah. And like I said, we never saw them have bacteremia with Ephicalis.
Mm-hmm.
So we don't know if it was because the Ephicalis couldn't translocate into the blood. I think that's less likely. I think any Ephicalis that ended up in the blood, the patient was able to like clear somehow, but for whatever reason, the faecium could not be cleared in this particular patient. Whether that's like a patient immune deficiency, we don't know, and we didn't study that here. But yes, in the time frame when the patient had Ephicalis, sort of relative dominance. The other thing is that it's relative abundance. And so what I didn't tell you about is like the absolute burden of Enterococcus in the patient's gut. But we kind of looked over time and didn't see a big shift. We didn't see like a decrease in Enterococcus in terms of CFU per gram of stool. So we think that there was a certain amount of space in this GI microbiome for Enterococci. And that space was either filled with epithelium or it was filled with Ephicalis. But yes, even though some people think of Ephicalis as having like a suite of virulence factors, in this particular patient, we didn't find any like association between, Ephicalis and feeling worse or having poor, poor clinical state. It seemed that at the time when they had Ephicalis dominant in their gut, they were not bacteremic with epithelium.
Yeah. So another thing that I was wondering was whether you tested any like control group of bacteria during that time period, like whether the population of E. coli or something like that. Like
we haven't. Yeah, we haven't looked. We do still have stool samples from all those time points, and we could do the full, shotgun for everything. I was telling somebody else earlier that I'm not a metagenomics person. This is as far into the metagenomics pool as I have gone, and I feel very uncomfortable doing metagenomics. I live in a world of single isolate. We know what it is. It's a pure culture, and I sequence it. But we have the samples, and again, we could look at broader community dynamics in this one patient. I mean, we are very limited in any conclusions that we can draw because it's one patient. But if we are able to treat more patients with Viriphage therapy, we know that the GI tract is usually the most abundant site of colonization. And so getting similar samples from other patients would help us, extrapolate and see if the findings are more broadly applicable.
Thank you.
I was really intrigued by those several cases of antagonism.
Certainly.
And I wondering, are you following it up? Do you have ideas? And actually, I was wondering how the phage infections were done to establish this pattern. to establish in your mind this pattern of antagonism?
Yeah, so the phenotype, to get into the details of the phenotypic screen, so Maddie and Kirsten, a technician in the lab, just plaqued the either single phage or combo on soft agar overlays.
Spot test.
Yeah, so, and they did serial dilutions. And so to call it active, they had to see single plaques. So simply clearing the lawn and inhibiting the lawn didn't count as active. They had to actually see titratable single plaques to call active.
What mechanisms?
Yeah, how could the antagonism work? I have no idea.
Yeah.
But it's certainly interesting. The activity assay is a little noisy, so we're kind of hesitant. They repeated it twice, and each person did their tests, and then they kind of sort of came together, and where they disagreed, they did it again. So we did the best that we could to try and assure ourselves that the results were reproducible. But they also would come to me and say like, oh, this plate looks kind of weird, or oh, I saw no inhibition at one dilution, and then I saw plaques at the next dilution. And there was a lot of weird stuff that went on in the screen. And I think now we're in the case, now we have to decide with all this screening data, like how do we follow up, and like what do we actually look for in trying to follow it up?
Maybe this is too off the wall, but I'm wondering if it would be helpful to try to find mutants of either one of those phages that could overcome the inhibition of.
Certainly. So we have selected in vitro phage resistant mutants that are resistance, when they're resistant to one, they also seem to be resistant to the other.
No, I didn't mean bacteria. I meant the one phage could overcome the antagonism of the other phage. And
I see.
Then sequencing might be in.
Certainly. Yeah. So we haven't played with like passaging them in vitro yet, but yeah, it is an exciting thing to think about.
And then since you have several different enterobacterial isolates that show this pattern, is there a bacterial host component?
Certainly. Yeah, that's a great idea.
Sure. I was just curious. So you mentioned that you had the Ephecalis strains. It was the same strain that sometimes had the drug resistance genes and sometimes didn't. If you were starting with pooled isolates of all the enterobacteria, depending on what kind of sequencing you did, I'm wondering if you could explain like your strategy of how you figured that out, because I could imagine that would be pretty tough to do.
So the vancomycin resistance in these bacteria is encoded on a plasmid. And so losing the resistance is simply losing the plasmid. And carrying this plasmid has an associated fitness cost that is small, but over time will compound. And so during the time periods when the patient was receiving vancomycin, during those times, we saw that the plasmid was relatively more abundant and there was more VRE. And then when they went off vancomycin, it wasn't immediate, but over time, the population probably loses this plasmid. And then we see, a shift of like the ephesium. We see fewer VRE fecium and more vancomycin susceptible fecium. But this is all based on the sequencing that we've done.
Cool. How did you know it was on a plasmid? Was it like already known? Or?
We know anecdotally, and from sequencing a lot of VRE from our setting, we know that for the Van-A operon, which is one flavor of vancomycin resistance, at least in our setting, and also more broadly in the US, it's almost always on a plasmid. Vancomycin resistance itself is always on a mobile element, but sometimes it's on a transposon that integrates into the chromosome. But in this case, I think it's always plasmid.
Awesome. Thanks.
Yeah.
Thank you so much. That was really, really fun to hear about. Really very interesting work. And as you can see, you got a ton of questions. So that was really fun.
Thank you all for your questions.
So yeah, are there any further questions? I don't mean to take over the floor too soon, but no, that's wonderful.
Yeah, I'll hang out after if other people have questions. Come and say hi. Yeah.
Yeah.
And thanks to everybody on Zoom for tuning in.
Yeah, that's right. Oh yeah, thanks to the Zoom.